Hello students, today we are going to discuss one more concept from sexual reproduction in angiosperms. The concept is microsporogenesis. Let's see the definition of microsporogenesis. The process of the formation and differentiation of microspores or pollen grains from microspore mother cells by reductional division is called microsporogenesis. So once recollect the structure of microsporangium. On maturation, the microsporangium produces archisporial cells. These archisporial cells undergo transverse division or periclinal division. So during this division, the cells which are formed towards outside are called as primary parietal cells, which are further divided to form 3 to 5 layers of microsporangial wall. They are endothecium, middle layers and tapetum. And the archisporial cells which are produced cells towards the inside are called as primary sporogenous cells. These primary sporogenous cells differentiates and to form pollen mother cells or microspore mother cells. These microspore mother cells involved in microsporogenesis. So let's see. Each microspore mother cell divides meiotically to form 4 haploid microspores or pollen grains, and they are remains arranged in tetrads. Let's see. Microspore mother cell which is in diploid condition, that is 2n condition. This microspore mother cell undergo meiosis division or reductional division to form 4 haploid microspores or pollen grains. They are in haploid condition. Now all these 4 microspores or pollen grains are arranged in tetrad. What is a tetrad? Tetrad is a group of 4 cells formed by meiosis from one diploid cell. Here it is clear, microspore mother cell is a diploid and it forms 4 haploid cells through meiosis division. The arrangement of pollen grains in a tetrad is affected by cytokinesis during meiosis. Now what is meant by cytokinesis? Cytokinesis is nothing but division of cytoplasm. The cytokinesis is of 2 types. They are simultaneous cytokinesis, successive cytokinesis. Simultaneous cytokinesis. The cytokinesis occurs simultaneously at the end of meiosis 2. We are all well knew that meiosis is occurred in 2 phases, meiosis 1 and in meiosis 2. Now this simultaneous cytokinesis is occurred at the end of meiosis 2. Let's see the photograph. So microspore mother cell undergo meiosis division. So in first meiosis division, 2 daughter nuclei are formed. These 2 daughter nuclei undergo meiosis 2 and finally it forms the 4 nuclei. After formation of these 4 nuclei, then it undergo cytokinesis to form the tetrad. So now this type of cytokinesis is called simultaneous cytokinesis because it is occurred at the end of meiosis 2. And the wall formation in this cytokinesis is centripetal. That means the wall initiation starts at peripheral walls and it is extended towards the center. This type of cytokinesis occurs in dicots. Successive cytokinesis. The cytokinesis occurs twice, that is, each of the two nuclear division is followed by wall formation. See the photograph. So microspore mother cells undergo meiosis division. The first meiosis division form the two nuclei, and these two nuclei are separated by the wall formation. That means cytokinesis is occurred after the end of meiosis 1 as well as at the end of meiosis 2. This type we can call as successive cytokinesis. And the wall formation in this cytokinesis is called as centrifugal, which means the wall initiation starts at the center and it is extended to the peripheral wall. This type we are called as centrifugal wall formation. And this successive cytokinesis occurs in monocots. Types of pollen tetrads. The factors influencing the types of tetrads are types of cytokinesis and the arrangement of microsporocytes. There are different types of pollen tetrads based on the factors. They are tetrahedral, isobilateral, linear, T-shaped, dehiscence. Let's see each one. Tetrahedral. It is formed by simultaneous cytokinesis. So which means the wall formation is occurred at the end of meiosis 2. See this photograph. In this 3 we are clearly visible, and fourth one it is mentioned as a circle at the center. That means the pollen grain is present the opposite side of that. And the example for this is dicots. Isobilateral tetrads. It is formed by successive cytokinesis. That means the wall formation is occurred at the end of meiosis 1 as well as at the end of meiosis 2. So this isobilateral tetrad is appears like this. Example, monocots. Linear pollen tetrad. It is due to the arrangement of microsporocytes. In a tetrad, the pollen grain is arranged linearly. This we can found in Halophylla. The fourth one, T-shaped pollen tetrad. It is also due to the arrangement of microsporocytes, and it is appears like this. Here there are 4 pollen grains. Follow the cursor, and these 2 transverse, and if you draw the line like this, it is appeared as a T. It is appeared. If you draw the line to follow the pollen grain like this, and it is appeared a letter T. That's why we are mentioning that the pollen grains are arranged in T shape. And this type we can observe in Aristolochia. And last one, dehiscence pollen tetrad. It is also due to the arrangement of microsporocytes, and it is appeared like this. And the fourth pollen grain in this photograph is present in the opposite side, and it is present in Magnolia. Unusual features in pollen tetrads. In Aristolochia elegans, all the 5 types of microspores arrangement are present. All 5 types means in this Aristolochia elegans we can found tetrahedral, isobilateral, linear, T-shaped, dehiscence. All 5 types are present. It is one unusual feature in this plant. And second one, in Drosera insectivorous plant, Typha, Hydrilla, etc. all the 4 pollen grains do not separate and thus form a compound pollen grains. So in those plants, Drosera, Typha, Hydrilla, in those plants, pollen grains are formed as a compound without separation. And third unusual feature in cypress, out of 4 pollen in a tetrad, 3 degenerate and 1 remains alive. So already we discussed during microsporogenesis, microspore mother cell undergo meiosis to form 4 haploid microspores or pollen grains. In that 4 haploid pollen grains, 3 are degenerate, 1 remains alive. That means one meiosis in this plant produces only 1 pollen grain. This is one unusual feature in this cypress. In Cuscuta, it is a stem parasite. More than 4 pollen grains are produced from one microspore mother cell. This condition is called as polyspory. That means the unusual feature polyspory we can observe in Cuscuta. In calotropes and in some orchids, all the pollen grains of an anther lobe remain united in a sac-like structure called as pollinium. So pollinium is formed in calotropes and in some orchids. In Schweitzeria, pollen grains occurs in dyads. So already we are discussed that the pollen grains are formed in tetrads. This is an unusual feature in Schweitzeria plant. The pollen grains are arranged in dyads. Dehiscence of anther. The process of dehiscence occur during stages of anthesis. What is meant by anthesis? It is the period during which a flower is fully open and functional. That period is called as anthesis. What is meant by anther dehiscence? So the final function of the anther that causes the release of pollen grains is called as anther dehiscence. The endothecium is responsible for anther dehiscence, that means to split the anther. Based on the direction of splitting, nothing but dehiscence, an anther releases pollen grain in the following ways. They are transverse dehiscence, longitudinal dehiscence, valvular dehiscence, poricidal dehiscence. Let's see each one in diagrammatic representation. Transverse dehiscence. See the photograph. So in this photograph, the anther is splits transversely and releases the pollen grains. This we can found in anthers of ocimum. So ocimum is commonly called as tulasi. Longitudinal dehiscence. In this photograph, the anther is splits longitudinally and releases the pollen grains. This we can found in anthers of datura. Valvular dehiscence. So in this photograph, the walls of anther splits and releases the pollen grains. This we can found in berberis. Poricidal dehiscence. So in this, the anther anterior region or tip region of anther splits like a pore and releases the pollen grains. And this we can found in solanum. Thank you. We can discuss one more concept in the next part.